Okay, so good afternoon, everyone. Welcome to the mini-school in May, an introduction to quantum biology. I'm very happy today to introduce to you Dr. Betony Adams. Betony did her master's and PhD at UKZN and is now a postdoc with us here in Stellenbosch. And she's been working for a while quantum effects in biological system, and particularly also on quantum effects that might play a role in our very own CPU, our brain. Yeah, and on the side, Betony also is active as a science communicator. Yeah, in the field of quantum biology and surroundings. Yeah. So, Betony, we are really very glad that you decided to offer a mini school on a topic that is very close to your heart and my heart. And I'm very much looking forward to listen to you. Yeah, you're welcome to share the screen. Yeah. And while you do that, maybe the usual reminder to the participant, please put your questions in the Q&A. And maybe at the end of the talk, we can give you the right to ask in person. Yeah. So thank you very much, Betony.
Can you see my screen?
Yes, yes, yes.
Yeah. Thank you so much, Francesco, for that introduction. I hope it is indeed an interesting mini school. And hello, everyone, and thank you for coming to this mini school. I'm really excited to tell you all about quantum biology, which I think is a really fascinating subject. In today's lecture, I thought it would be useful just to start by bringing up some of the main concepts and terminology that you might encounter in quantum biology, both from the physics and the biology point of view. I won't be able to go into any great detail as there's a lot to get through, but hopefully it will give you the framework to go and explore further. I'll talk for about 45 to 50 minutes to give you some time for questions at the end. So this is a mini school on quantum biology. So what exactly quantum biology is still an open question. Is it just another iteration of biophysics, or does it deserve a category of its own? I won't waste too much time on this debate, but I'll rather tell you why I think it is really interesting. And I want to start by sharing some of my favorite biology facts. So quantum biology looks at things like photons, electrons, and protons in the materials that make up living organisms, like proteins or fats or cells or even the brain. So you might think this is a terrible oversimplification of the complexity and diversity of nature, which is an argument which has been raised against quantum biology. So to start this presentation, I thought it would be interesting to see whether there are any fundamental similarities between biological systems on the microscopic scale. So on the left-hand side of the slide is a simple single-celled organism, a bacteria. It has a cell body, and then it has those hairs that stick out like a tail called flagella that it uses to respond to environmental cues and move towards and away from things. So flagella are powered by membrane potentials, which just means the concentration of charged ions across the cell membrane. And this concentration can be changed by opening pores in the cell membrane to let ions go in or out. And altogether, it's a mechanism for sensing and reacting to environmental influences. And on the right-hand side is another single cell, but this is a nerve cell that collects into groups and makes up the brain, which is maybe the most astonishing of biology's creations. On a mechanistic level, however, a single neuron is not so much different to the bacteria. In response to a stimulus, pores called ion channels open and change the membrane potential. And if this potential gets high enough, then the nerve fires and a signal is sent down the long tail of the nerve, that is called the axon, before being processed and resulting in some sort of response to the stimulus. So we might consider ourselves very far removed from a bacteria, but on the microscopic level, there are some quite profound similarities. Systems is that they are both discrete or separate from the environment, but also open to this environment, and they extract energy from the environment. And to do this, they have developed membranes to control the border between outside and inside. For simple organisms, one of the main sources of energy is, of course, the sun. So how did they evolve to manage, use, and store the sun's energy? Well, one of the ways they achieve this is by turning this energy into a membrane potential. And they do this by using light to open channels and control the flow of negative or positive charged ions, such as sodium, potassium, and calcium, across membranes. In effect, they create a potential difference across the membrane where one side is more positive and the other side more negative. And how do they do this? Well, they use these specific light-sensitive molecules called chromophores. Chromophores are molecules that absorb invisible wavelengths, and they are responsible for a lot of the color that you see in nature. And chromophores are also at the heart of many of the topics in quantum biology. Photosynthesis, for example, is preoccupied with chromophore chlorophyll, which makes things green. The radical pair hypothesis of bird migration is thought to depend on cryptochrome, which is blue light absorbing. And another really important chromophore is retinal. So when retinal absorbs light, it undergoes an electron transfer that is linked to a conformational change, which opens an ion channel and allows ions to flow through the membrane and change the membrane potential. And one of the results of this is that this potential can be used to power hairs, like the bacteria, these flagella, that cause the organism to swim towards or away from the light. As these first organisms evolved, it is thought that they began to organize retinal in one spot of the cell membrane to maximize the effects and better control the flagella. And this concentration of retinal chromophores on the cell is called an eye spot. And you can see them in red in the picture of the cells on the left. And the reason I think this fact is so amazing is because we are not actually so far removed from these very, very distant evolutionary ancestors. We also have a concentration of retinal chromophores that absorb light and change and cause a change in ions across membranes, in particular across nerve cell membranes, causing nerves to fire. And we call these concentrations of retinal chromophores our eyes. We use them to mediate the border between our inside and outside. We also turn light into a signal that prompts our behavior. An even more fundamental question in the context of quantum biology is, what is life? There are lots of theories about how life began, which I won't get into here. But to have life, you first need the molecules that life is made from. So in 1952, the Miller-Urey experiment, which is a very famous experiment, showed that you could put a collection of prebiotic molecules like water, methane, ammonium, and hydrogen into a flask and then send electricity through this flask. And what you get is the amino acids that are essential to building the proteins that sustain life. Some theories suggest lightning may have played the role of the spark at the beginning of life, but what about in places where there was no lightning, such as deep sea vents? Were there some localized sparks? There are some theories that iron sulfur compounds, such as iron pyrite, which you may have heard of as fool's gold, could have acted in this way because it is redox active and facilitates electron transfer. I really like the thought of this, because if it's true, it means that fool's gold is infinitely more valuable than gold. But it also means that electron transfer might have been implicated in the very beginning of life. And charge transfer, the movement of electrons and protons, is still extremely important in biological systems and forms the foundation on which quantum biology is built. So before we get into basic concepts, let's have a quick look at the history of quantum biology, which is quite closely tied to the history of quantum mechanics in general. The origins of quantum mechanics go back to the earlier 20th century, when classical physics could no longer explain certain experimental results. So in 1900, Planck introduced the idea that energy is quantized, proposing that it is emitted in discrete packets called quanta, laying the foundation for quantum theory. Einstein built on this in 1905 by explaining the photoelectric effect using quantized light particles, or photons, and providing strong evidence for the particle nature of light. In 1913, Bohr applied quantum ideas to the atom and suggested that electrons orbit the nucleus in specific quantized energy levels. Compton further supported the particle theory of light with his 1923 experiment, that showed that photons could scatter off electrons like particles. In 1924, de Broglie proposed that particles, like electrons, also have wave-like properties, introducing the concept of matter waves. And this led Schrödinger to the development of wave mechanics, where he formulated an equation that describes how quantum systems evolve over time. And around the same time, Heisenberg created matrix mechanics and introduced the uncertainty principle, which states that certain pairs of properties, like position and momentum, cannot be precisely known at the same time. This is a very simplified version of the origins of quantum mechanics, but together these scientists and others revolutionized our understanding of the microscopic world and laid the groundwork for modern quantum mechanics. So quantum biology is often thought of as being a relatively recent field of research, but the reality is that the founding figures of quantum theory were already thinking about how it might give some insights into the natural world and living systems. Schrödinger, who's famous for his wave equation and his cat thought experiment, wrote a famous book called What is Life? And Bohr, who updated the model of the atom to include quantum mechanics, gave a famous lecture called Light and Life. Light was central to the development of quantum mechanics, and the first ideas of quantum mechanics were the result of Planck trying to explain blackbody radiation and Einstein trying to explain the photoelectric effect, both of which deal with the interaction of light with matter. Biological systems are also made up of matter, and light is fundamental to life, most obviously in the fact that plants turn light into biomass. They photosynthesize. Schrödinger's 1944 What is Life and Bohr's 1932 lecture Light and Life both explore how the principles of quantum mechanics might apply to living systems. In What is Life, Schrödinger writes about the molecular basis of heredity, and he asks how the order and stability of genetic inheritance could arise out of the disorder of thermodynamics. And he suggested that the genetic information must be stored in what he called an aperiodic crystal, which is a term that inspired future discoveries of the DNA double helix. In his Light and Life lecture, Niels Bohr questioned whether it would be possible to describe living processes purely in terms of physics and chemistry. And the story goes that the physicist Max Delbruck was in the audience when Bohr delivered his lecture, and it inspired him to switch from physics to biology. And Delbruck went on to pioneer molecular biology. What I also found really interesting is that Bohr gave this lecture, The Light and Life, at an International Congress on Light Therapy. I'm not sure how many of you know that the 1903 Nobel Prize in Medicine was awarded to Niels Ryberg-Finsen for his use of light therapy to heal lupus vulgaris. And yet, despite this very long history of interest in the interaction of light with living matter, quantum biology is still considered by many to be a new field of research. So let's look a bit more closely at some of the main events in the history of quantum biology. So before he gave his Light and Life lecture, Bohr delivered a lecture titled The Quantum of Action and the Description of Nature at the University of Copenhagen. The lecture explored the nature of quantum mechanics and its implications for the description of the physical world. In 1932, Pascal Jordan published the first paper in which quantum biology is referred to by name, and then in 1941, he published the book Physics and the Secret of Organic Life, in which he questioned whether the laws of quantum physics were fundamental to life processes. This book was followed shortly in 1944 by Schrödinger's What is Life, which, as I've already mentioned, inspired Watson, Crick, and Franklin's discovery of DNA's double helix. In 1963, Loudon hypothesized that quantum tunneling might be the mechanism for DNA point mutations. And in 1966, we have the first experimental verification of quantum biology, when DeVault and Chance observed tunneling in enzymes, which in 1974 was developed by Hopfield in a theoretical context. While it's not explicitly on the timeline, I would also mention the development of spin chemistry in the 60s and 70s, which is a field of research which looked at how magnetic fields could influence chemical reactions. And from spin chemistry, we get the radical pair mechanism, which Schulten hypothesized in 1976 could be the mechanism by which birds navigate using the Earth's magnetic field. And birds were first observed to use the geomagnetic field in the 70s by the Wiltshires. In 1989, Judith Klinman's Berkeley group observed proton tunneling in enzymes, and in 2000, Torsten Ritz suggested that cryptochrome could be the site of the radical pair mechanism of bird magnetoreception. And then from the 2000s onward, experiments also began to show evidence for quantum coherence and energy and charge transfer in photosynthesis, though whether this is excitonic or vibronic coherence is contested. This is not an exhaustive list of the progress of quantum biology, which has accelerated in the last decade or so, but it does give you some idea of the long history that quantum biology has, despite it often being referred to as a new field. So in this introductory lecture, I thought it would be useful to go over some of the basic concepts and terminology of quantum biology. Because it is an interdisciplinary field, it marries physics with biology, it can be quite challenging for both physicists and biologists who have never encountered certain ideas or terms before. I don't have enough time to go into anything with too much detail, but I've tried to cover some of the most often mentioned ideas, first from the point of view of physics for biologists, and then basic biology for physicists. So in quantum mechanics, one of the things you will hear over and over again is this idea of a quantum system. So what exactly is a quantum system? Well, defined formally, a quantum system is any physical system that is governed by the laws of quantum mechanics, which is the branch of physics that describes the behavior of matter and energy at the smallest scales, typically at the level of atoms, electrons, photons, and other subatomic particles. More intuitively, you might think of the quantum system as the thing that you are interested in looking at. So, for example, if any of you did physics at school, you probably did many worked examples on the movement of blocks down slopes or cars accelerating along roads. In these questions, the system was the block or the car, for example. Quantum systems are just smaller things of interest, such as photons, phonons, protons, neutrons, or electrons. For those of you not familiar with what these things are, I'll briefly give you some context. So one of the first major accomplishments of quantum theory was Bohr's model of the atom in 1913. And in this model, electrons orbit the nucleus, which is made up of positively charged protons and neutral neutrons, in specific fixed energy levels or shells, rather than spiraling inward, which is what classical physics would predict. Bohr suggested that electrons could jump between energy levels by emitting or absorbing a quantum of energy or photon, and this emission and absorption of light explains the discrete lines that are seen in atomic spectra, which are like the light equivalent of a fingerprint for different types of matter. So why is this important to quantum biology? Well, it tells us the fundamental way in which light interacts with matter, which you will see is a big part of quantum biology. It also tells us about the movement of electrons, which is arguably at the very heart of living systems. And the Nobel Prize winner Albert Szent-Györgyi once wrote that life is nothing but an electron looking for a place to rest. So if a quantum system is the thing that we are interested in, what is a quantum state? So in the classical physics examples like the block or the car, we are interested in what interesting properties these things have, like position or acceleration. And it's the same for quantum systems. If the things we're interested in are, for example, electrons maybe being repelled by each other, then the state describes properties of these electrons, such as position, momentum, energy, etc. Unlike classical systems, however, a quantum system does not have definite values for properties like position or momentum until they are measured. Instead, it can exist in superposition of all possible states. In quantum mechanics and quantum biology, you will see these symbols psi and rho quite often. They are mathematical representations of the state of a system. Psi represents a state vector, and it contains all the information needed to predict the outcomes of measurements on the system, such as position, momentum, spin, or energy. The state vector exists in an abstract mathematical space called Hilbert space. Why do we need a vector? Well, a vector can represent a superposition of multiple possible states, meaning a system can exist in a combination of different configurations until it is measured. When a measurement is made, the state vector collapses to one of the possible eigenstates associated with the measured value, and the probability of each outcome is given by the square of the amplitude in the state vector. This probabilistic nature of quantum mechanics makes the state vector a uniquely non-classical concept, and distinguishing quantum theory from classical physics, where systems have definite properties. Rho is the density matrix, which can be thought of as a generalization of the state vector. The density matrix or operator is a mathematical tool used to describe the state of a quantum system, especially when the system is in a mixed state rather than a pure state. A density matrix can represent both pure states and statistical mixtures of different quantum states, which occur when there's uncertainty or lack of complete knowledge about the system. So another term you will hear over and over again in quantum biology is the Hamiltonian. In quantum mechanics, the Hamiltonian is an operator that represents the total energy of a system, including both kinetic and potential energy. I should have explained in more detail what an operator is, but very simply, if the state vector or density matrix contains all the information about a system of interest, including superpositions, then an operator is how you get a specific piece of information out of the state vector. For example, the position operator gives the position distribution of a particle, while the momentum operator gives its momentum distribution. Most quantum operators are Hermitian, meaning that they have real eigenvalues and correspond to physically measurable quantities. Quantum operators are like the bridge between the abstract maths of quantum theory and the real world outcomes that we want to observe and measure. The Hamiltonian operator plays a central role in also in determining how the quantum system evolves over time. And its eigenvalues correspond to the system's possible energy levels, making it essential for calculating observables and predicting measurement outcomes. So some common Hamiltonians that you will see in quantum biology is the two-level system, which can describe any quantum system that can exist in two different energy states, such as an electron with a ground or excited state, or a particle with a spin up, spin down. Another common Hamiltonian is the harmonic oscillator, and because this Hamiltonian can model the vibrational modes of molecules, it is a useful Hamiltonian for representing the environment of a system in quantum biology. And another Hamiltonian we will be seeing a lot more of in these lectures is the radical pair Hamiltonian, which describes each of the possible spin interactions a pair of electrons can have. So once you have the Hamiltonian describing the system of interest, what do you do? Well, a system that stays the same is not going to be that interesting or tell you that much. So we're interested in how systems change over time, which is often called the time evolution of the system. And the Schrödinger equation is the foundational equation of quantum mechanics that describes how the quantum state of a physical system evolves over time. In its time-dependent form, it is written as the time derivative of the state vector equal to the Hamiltonian acting on the state vector. This equation plays a role similar to Newton's laws in classical mechanics, but for the probabilistic world of quantum particles. The Schrödinger equation tells us how the state of a particle or system evolves with time and what outcomes are possible when we measure its properties. And to solve this equation, we use the unitary time evolution operator, which is an operator that tells us how the quantum state evolves over a closed system, over time in a closed system, and it's denoted by U at t, and its role is to evolve an initial quantum state at time 0 to its state at time t. But Schrödinger's equation describes how ideal quantum systems evolve in time. And by ideal, I mean quantum systems that are isolated at low temperatures and don't interact with their surroundings. But biological systems are necessarily interactive. No living organism can avoid interaction with its environment, never mind the fact that biology conventionally operates at warmer temperatures. In this context, the Schrödinger equation will not be an accurate description of how an electron, for example, behaves in the context of biology, in which there are a million things that this electron can interact with. To have a more accurate description of the electron's behavior, we need open quantum systems theory. Open quantum systems are quantum systems that are modeled to interact with an external environment or surroundings, as opposed to closed systems, which are isolated. The interaction with the environment leads to effects such as decoherence and dissipation or loss of information, which means the system's evolution is no longer purely unitary and means it cannot just be described by the Schrödinger equation and a unitary evolution operator. Instead, the combined system of the combined state of the system and the environment is evolved unitarily, and then we trace out the environment to look at the evolution by itself. So instead of the state vector and the Schrödinger equation, the state of an open quantum system is often described by a density matrix, and its evolution follows more general equations such as master equations. And a typical master equation for an open system will have a term describing the closed dynamics and a dissipated term which describes the effects of the environment. The closed dynamics, or the unitary part, represents the system's ideal evolution if it was isolated, and it includes the system Hamiltonian. whereas the dissipator part accounts for the interaction with the environment and includes effects such as decoherence, which is the loss of quantum superpositions, and dissipation, which is energy exchange with the environment. So when you hear about master equations, you might also hear some discussion of Markovian or non-Markovian effects. Markovianity describes memory effects, or how information from the system is lost to the environment. A Markovian master equation describes the time evolution of an open quantum system when memory effects from the environment are negligible, meaning that the environment does not remember past interactions with the system. This leads to a simplified memoryless evolution where the system's future state depends only on its present state, not its history. And the most general Markovian master equation, which you will see a lot of in quantum biology, is the Gorini, Kosakowski, Sudosh, and Lindblad equation, which you will often just hear called the Lindblad equation, where HS is the system Hamiltonian, or H in the commutator. LK are the Lindblad operators that describe how the environment interacts with the system. The first term gives you the coherent quantum dynamics, and the second term gives you the dissipator. Non-Markovian master equations, on the other hand, describe the evolution of open quantum systems where memory effects from the environment cannot be ignored, meaning the environment retains and can later feed back information into the system. Unlike the Markovian case, non-Markovian dynamics can account for delays, recoherence, and complex system environment correlations. I'm not going to go into detail here. I think Dr. Graham Pleasance has given some lectures on open systems, which should be on the NITheCS website, and he's far more of an expert than me. For a very basic overview, here's a table comparing Markovian and non-Markovian master equation approaches. In terms of memory effects, Markovian approach has no memory. The system's evolution depends only on its present state. In non-Markovian approaches, it includes memory, so the system depends on past history. For environmental correlation, the Markovian approach has fast decaying path correlations. For the non-Markovian approach, slow decaying or structured correlations. An example equation for Markovian approaches, as I've mentioned, is the Lindblad master equation. For non-Markovian approaches, you may hear people talk about the Nakajima-Zwanzig equation, time convolutionless, or the hierarchical equations of motion. In terms of accuracy, the Markovian approach is approximate, but it is valid for specific conditions. The non-Markovian approach is often more accurate, especially for strong coupling or structured parts. However, the trade-off is that for Markovian approaches, the computational cost is much lower than for non-Markovian approaches, which have to take into account memory tracking or extra auxiliary terms. And then some physical examples of of systems that use or are suited to Markovian approaches are qubits in a thermal bath, optical cavities or atomic decay, and non-Markovian approaches, photosynthetic complexes. You'll often see that use the hierarchical equations of motion, solid-state qubits or spin boson models. I mentioned very briefly how to solve the Schrödinger equation, but how do we solve these more complicated master equations in the context of quantum biology? Because it is dealing with more complex systems, quantum biology depends quite strongly on numerical solutions. And in the past few years, a number of useful tools have emerged for computing solutions to quantum biology problems. I don't have time to go into the details of these tools, but there are excellent tutorials available online. I've mentioned here some of the most popular examples. If you look them up, they're well documented. Quantum Toolbox in Python, or Qubit, is open source software for simulating the dynamics of open quantum systems, and there are some very nice tutorials on quantum biology examples. Radical Pi is a toolbox specifically developed for radical pair spin dynamics, as is MolSpin, an open source general software designed to perform any kind of calculations on spin systems. So a recurring topic in the effort to define what quantum biology is this distinction made between trivial and non-trivial quantum effects. And if you've followed quantum biology at all, you may have heard people talking or arguing about whether you can even make this distinction. For my purposes, I take trivial quantum effects to mean those effects that apply to all matter, whether living or non-living. So all atoms are described by the laws of quantum theory. And understanding this does not necessarily tell us anything new about living systems. On the other hand, as I understand it, non-trivial quantum effects are those effects that are implicated in the functionality of biological systems. This may be something as simple as adding a neutron to an atomic nucleus that in turn has a pivotal effect on, say, consciousness. The triviality doesn't refer to the quantum effect as much as it refers to the biological outcome. And some examples of the quantum effects that play a non-trivial role in biological function are superposition, coherence, tunneling, and entanglement, which I will now give brief introduction to. So the superposition principle is arguably at the heart of quantum mechanics. Many of you may be familiar with it from Schrödinger's famous cat thought experiment, where a cat is placed in a sealed box with a radioactive atom and a vial of poison that breaks if the atom decays and thus kills the cat. So the radioactive atom exists in a superposition of decayed and not decayed until observed. And since the cat's fate is tied to the atom, the cat is also in a superposition, both alive and dead, until someone opens the box and observes it. While it may seem counterintuitive when applied to cats, superposition follows naturally from the Schrödinger's wave equation, because linear combinations of solutions to the Schrödinger equation are also solutions to the Schrödinger equation. Superposition is well described in the context of superimposed waves and results in interference effects, which depend on how coherent the waves are. So in the quantum context, coherence thus describes the relationship between states in a superposition. Quantum tunneling is a phenomenon in quantum mechanics where a particle can pass through a potential energy barrier, even if it doesn't have enough energy to overcome it classically. And this happens because particles in quantum theory are described by wave functions, which can extend into and beyond classically forbidden regions. Although the probability of finding the particle inside the barrier is low, it's not zero, meaning there's a chance it could be found on the other side of the barrier. And quantum tunneling is essential in many physical processes, such as nuclear fusion in stars, alpha decay, and the operation of modern technologies like scanning tunneling microscopes. Quantum entanglement, which Einstein apparently called spooky action at a distance, is a fundamental phenomenon in which the quantum states of two or more particles become linked such that the state of each particle cannot be described independently of the others, even when separated by large distances. Measurements on one particle instantaneously affect the outcome probabilities of measurements on the other in a way that violates classical expectations. And Bell's inequalities, which you might hear about, were derived to test whether the predictions of quantum mechanics, especially regarding entanglement, could be explained by any local hidden variable theory. And experiments have repeatedly suggested that Bell's inequalities are violated, confirming that nature follows quantum mechanics and that entanglement involves non-local correlations that defy classical explanations. One of the main arguments against quantum effects playing a role in biology is decoherence, which describes the loss of the interesting quantum effects that I've just told you about. So if you've read any quantum biology paper, you will probably have heard the phrase describing biology as being too warm, wet, and messy for quantum effects to survive on relevant timescales. Quantum effects are fragile. They're normally observed at very low temperatures because at higher temperatures, the environment around the quantum system is moving too much and interacting too much with the system, causing decoherence. For example, an electron transfer in biology is happening within a protein, which is vibrating at various different frequencies. But some people have suggested that actually this environment around a quantum system might be helpful. Nature is a very clever engineer. I think one of the clearest examples of this is how we see different colors. So in our eye, we have three types of cone cells. Cone cells allow us to see color. Cone cells work when light activates the light-sensitive chromophore called retinal, which I've already told you about, and it causes an electron transfer. But if retinal only absorbs light at one specific frequency, how do we see light of different frequencies? Well, the trick is the protein. The protein that the retinal is embedded in is different. Opsin proteins all have the same chromophore, retinal, but they have different protein structures. And the protein environment tunes the quantum system in a favorable way. So perhaps biology has evolved to harness the movements of proteins to facilitate rather than disrupt quantum effects. So that concludes the introduction of physics concepts. I'm sorry if I had to go through them so fast. You will hear some more about these ideas in the upcoming lectures, but hopefully this gave you some idea of the basic tools you need for doing quantum biology from a physics point of view. But before we get too preoccupied with quantum objects of interest, such as single electrons, I thought it would be useful to get some context on where this physics is happening, the biological context. In biology, a cell is the basic structural and functional unit of all living organisms. It is the smallest unit of life that can carry out all the processes necessary for life, including growth, reproduction, and energy conversion. Cells come in two main types: prokaryotic cells, which lack a nucleus, like bacteria, and eukaryotic cells, which have a nucleus and other specialized structures called organelles, like those in plants, animals, and fungi. Key components of a typical cell include the cell membrane, which controls what enters and leaves the cell, cytoplasm, which is a jelly-like fluid where reactions occur, the nucleus, which stores genetic information. Some cells, like plant cells, also contain chloroplasts for photosynthesis and a cell wall for extra support. And all life, from single-celled organisms to complex multicellular creatures, is built from cells. Within multicellular organisms, cells can be highly specialized. For example, nerve cells transmit electrical signals, while muscle cells contract to enable movement. And one important type of cell is the stem cell, which is undifferentiated and has the unique ability to divide and develop into various specialized cell types. This makes stem cells crucial for development, repair, and regeneration. Other cell types include blood cells, epithelial cells, and immune cells, each playing a vital role in the function of the organism. Despite their differences, all cells share common features like cell membrane, cytoplasm, and genetic material. In multicellular organisms, like humans or animals or plants, cells group together to form tissues, which then combine to form organs like hearts, lungs, or leaves. And then organs work together in organ systems like the digestive system or circulatory system to carry out complex functions. And together, these systems make up the whole organism. So I would like to focus on a few specific structures in cells because you will be hearing more about them in the context of quantum biology. Mitochondria, if you did biology at school, you will remember they are often called the powerhouses of the cell because they are responsible for producing the energy that cells need to function. However, they're also implicated in a number of other functions, not least the generation of reactive oxygen species. These small organelles are found in the cytoplasm of almost all eukaryotic cells, and they generate energy in the form of adenosine triphosphate, or ATP, through a process called aerobic respiration. Mitochondria have a unique double membrane structure, an outer membrane that protects the organelle, and a highly folded inner membrane where the electron transport chain takes place. These folds, called cristae, increase the surface area for energy production. And what is really interesting is that mitochondria have their own DNA, which suggests that they may have evolved from ancient bacteria in a symbiotic relationship. And they are especially abundant in cells with high energy demands, such as muscle and nerve cells. And they play a key role in metabolism, cell signaling, and even cell death. Chloroplasts are specialized organelles found in plant cells and some algae, and they are the sites where photosynthesis takes place. These green disc-shaped structures contain the pigment chlorophyll, which I've told you about. It captures sunlight and uses its energy to convert carbon dioxide and water into glucose and oxygen. Like mitochondria, chloroplasts have a double membrane and contain their own DNA. Chloroplasts are essential not only for making food in plants, but also for producing the oxygen that most living things rely on to survive. The cell nucleus is a large membrane-bound organelle found in most eukaryotic cells, and it serves as the control center of the cell. It houses the cell's genetic material, or DNA, deoxyribonucleic acid, which carries the instructions needed for building proteins and directing all cellular activities. The DNA is organized into long structures called chromosomes, and the nucleus is surrounded by a nuclear envelope, which is a double membrane with pores that allow materials like RNA and proteins to move in and out. So in the physics section, we talked about electrons, protons, and neutrons as the materials out of which atoms are made. All matter is made out of atoms, but what are the important materials out of which life is made? Biological materials are carbon-based, and any of you who are familiar with organic chemistry will recognize some of these molecules. Biological materials fall into four main categories, which I'll go over very briefly. Nucleic acids, such as DNA and RNA, are made out of elements carbon, hydrogen, oxygen, nitrogen, and phosphorus, and their function is to store and transmit genetic information. Proteins are chains of amino acids made predominantly out of carbon, hydrogen, oxygen, nitrogen, and sulfur, and their function is a variety of functions, which I'll get into just now, but structure, they make up enzymes, they're used in signaling and transport. Lipids are fats, oils, and membranes made up of the elements carbon, hydrogen, oxygen, and then phospholipids have phosphorus. And their function is energy storage, insulation, and they make up cell membranes. And then finally, carbohydrates or sugars and starches made of carbon, hydrogen, and oxygens. They provide energy and structural support such as cellulose and cell walls. Out of these materials, you will hear more about some in quantum biology, so I thought it would be useful to give you a few more details about them. First up is DNA and RNA. DNA, deoxyribonucleic acid, and RNA, ribonucleic acid, are nucleic acids that store and transmit genetic information in living organisms. DNA carries the long-term information about organisms with its sequence of nucleotide bases, adenine, thymine, cytosine, and guanine, predominantly encoding the instructions for making proteins. When a protein needs to be made, a section of DNA is transcribed into messenger RNA, or mRNA, which is a temporary single-strand copy of the relevant genetic code. This then travels to the ribosome, where it undergoes translation. Its sequence is read in triplets or codons, each corresponding to a specific amino acid. Transfer RNA and brings the correct amino acids in sequence and allows the ribosome to assemble them into a polypeptide chain, which folds into a functional protein. DNA and RNA work together in this regulated process that converts genetic information that has been stored over the lifetimes of species into proteins that are essential for life. So one of the most interesting of the biological materials, in my view, are proteins. Proteins are large, complex molecules made up of chains of amino acids that play essential roles in nearly all biological processes. The sequence of amino acids determine protein's unique three-dimensional folded structure, which in turn dictates its function. Proteins play many roles in cells, including acting as enzymes to catalyze chemical reactions, structural components that provide support, like collagen and connective tissue, transport molecules such as hemoglobin that carries oxygen, hormones and antibodies in the immune system, and their diversity makes proteins the primary tools and machinery of life. In the context of quantum biology, proteins also offer a dynamic scaffold that may enhance or protect quantum effects. So another key biological molecule in quantum biology is the enzyme. Enzymes are specialized proteins that act as biological catalysts, speeding up chemical reactions in living organisms without being consumed in the process. Each enzyme has a unique active site, which is a region where specific substrate molecules bind and undergo chemical transformation. And the lock and key model has conventionally been used to explain how enzymes achieve this specificity. The enzyme's active site, or the lock, has a precise shape that fits only a particular substrate, the key, ensuring that enzymes catalyze only specific reactions. And you will hear much more about this lock and key mechanism in later lectures. Once the substrate binds, the enzyme can lower the activation energy required for the reaction to proceed, allowing it to happen much faster. You will also hear a lot about ligand-receptor interactions in later lectures. Ligands are molecules that bind to specific receptors, typically proteins located on the surface of or inside cells, to initiate a specific biological response. This interaction is fundamental to cell communication and signal transduction, allowing cells to respond to changes in the environment. Ligands can be hormones, neurotransmitters, drugs, or other signaling molecules. When a ligand binds to a receptor, it often triggers a conformational change in the receptor that activates a downstream signaling pathway. One major class of receptors you will hear about is G-protein coupled receptors, or GPCRs, which span the cell membrane and activate internal G proteins when they are bound by a ligand. GPCRs are involved in a wide range of physiological processes, including vision, smell, mood regulation, and immune response. Other types of receptors include ion channel receptors, enzyme-linked receptors, and intracellular receptors, which each play critical roles in mediating signals that control everything from gene expression to muscle contraction. Another very important class of biological molecules, which I have told you a little bit about in the context of quantum biology, are chromophores. If quantum biology is about the interaction of light with living matter, then chromophores are what facilitate this interaction. Chromophores are the parts of molecules responsible for their color, absorbing specific wavelengths of light due to the presence of conjugated systems, which are regions where electrons can delocalize across alternating double and single bonds. When light hits a chromophore, the energy can excite electrons from a lower to a higher energy level, and the wavelength of light absorbed determines the color the compound appears to the human eye. In biology, chromophores play essential roles in processes like photosynthesis, chlorophyll, vision, retinal, and UV protection, like melanin. Some proteins, such as fluorescent proteins, contain intrinsic chromophores that absorb and emit light, making them valuable tools in imaging and molecular biology. And in quantum biology, you will hear a lot about chromophores such as tryptophan and the protein scaffolds they are embedded in. Signaling mechanisms are central to biological systems, and one of the most important of these mechanisms are reactive oxygen species, which are highly reactive molecules containing oxygen, including superoxide, hydroxyl radical, and hydrogen peroxide. While they are often associated with cellular damage and oxidative stress, ROS also play essential roles as signaling molecules in various biological processes, including cell growth, cell death, and immune responses. At controlled levels, ROS regulates signaling pathways involved in inflammation, but excessive levels can damage DNA, proteins, and lipids and contribute to chronic inflammation and diseases such as cancer, cardiovascular disease, and neurodegeneration. And cells use antioxidant systems to balance ROS levels and prevent this oxidative damage. You will hear quite a bit about reactive oxygen species in quantum biology. There are also other important reactive species, such as reactive nitrogen species, but the role of these in quantum biology is less well developed. Quantum biology is strongly focused on electromagnetic effects in biological systems. And we've talked about electrons, but what other ways do cells generate electrical degrees of freedom? Well, ions are charged particles, such as sodium, potassium, and calcium, that play crucial roles in generating and maintaining the membrane potential, which is the electrical voltage difference across the cell membrane. Membrane potential is vital for various cellular functions. In nerve cells, rapid changes in membrane potential trigger action potentials, which are electrical impulses that transmit signals along neurons. In muscle cells, these lead to contraction. In mitochondria, ion gradients, especially of protons, across the inner membrane create an electrochemical gradient known as the proton motive force, which drives ATP production. Additionally, ion flows and membrane potential influence tissue regeneration and cell migration. Electron transport chains play a key role in quantum biology for a number of reasons, because they combine electron transfer as well as the production of radical pairs and reactive oxygen species, all of which are topics of interest in quantum biology. You will be hearing more about them in upcoming lectures, but I wanted to introduce them quickly here. So electron transport chains are a series of protein complexes and electron carriers embedded in membranes, such as the inner mitochondrial membrane, that play a central role in cellular respiration and photosynthesis. And their primary function is to transfer electrons from high energy molecules like NADH or FADH, or photosystem generated electrons in photosynthesis through a series of redox reactions. As electrons move along this chain, energy is released and it's used to pump protons across the membrane, which creates a proton gradient. And this gradient drives ATP ATP synthase, which is a molecular machine that synthesizes ATP, and ATP is the main energy currency of the cell. And finally, terms such as respiration and photosynthesis and metabolism keep coming up. And for a long time, I was confused about exactly how these biological functions were related to each other. I thought it would be useful to finish our biology section on a discussion of the centrally important concept. Metabolism is the set of all chemical reactions that occur within living organisms to maintain life. These reactions are broadly divided into two categories, which is catabolism, breaking down molecules to release energy, such as glucose breakdown and respiration, and anabolism, which builds up complex molecules from simpler ones, such as protein or DNA synthesis or biomass production and photosynthesis. And then the energy released from catabolic reactions is often stored in the form of ATP, which is then used to power anabolic processes and other cellular functions. Metabolism is highly regulated and interconnected, involving a number of enzymes and pathways that allow cells to respond to their environment, to grow, to reproduce, and to maintain homeostasis. Metabolism is also bound up with information and signaling processes in cells. I'm going to stop here to allow some time for questions. But next lecture, we will look more closely at specific quantum effects that I've mentioned briefly here and what biological functions they possibly play a role in. So thank you very much for listening. That's just some image references.
Yeah, thank you very much, Betony. Betony, can you please switch off the speaker there? I switched on here. Thank you very much. Are there questions for Betony? I see we have a participant with raised hand, and I'm very happy to allow to talk to both of them. Hello, George, and hello, Mbuzo. Please, Mbuzo, I think your hand was up first. You're welcome to ask.
Sorry, are you speaking? You're speaking to me?
Yes, please, George, please go ahead. Yeah.
Okay, thank you. Firstly, that was a brilliant lecture. Thank you very, very much. I've got a question and a comment. The question is, very early on you mentioned phonons, and I was rather surprised that you mentioned phonons. In what context in biology do phonons play a role?
So mainly in the vibration-assisted tunneling. So where the protein the vibrations of the protein environment might facilitate electron transfer reactions. As far as I understand it, that's the main place.
Okay, I'd love to hear more about that. The second point is that you mentioned you gave the standard story about the Schrödinger cat. Now, I gave a talk at NITheCS quite a while ago where I denied that there could possibly be a wave function for a cat. I think it's a fiction that it is not possible to have a wave function for a cat or a brain or anything like that. And I could go into that in detail, but I simply don't believe. You often see this so-called equation, psi cat equals A psi alive and B psi dead. And I think that is total nonsense. There is no such thing as a wave function for a cat.
Yeah, I was using it more as I think that's probably the main, if people aren't familiar with the quantum physics, that's probably the place they've heard Schrödinger. So it was more just as an introduction to Schrödinger. But yeah, I think that wasn't that the point of the thought experiment, to illustrate how ridiculous it is applied to, or how counterintuitive it is applied to classical objects. But
it was, but it's got into a common usage as if it actually happens, and I think that's completely wrong.
Well, I'll direct people to your talk, hopefully, to go and hear more about it.
Thank you.
Thanks.
I think Mbuzo raised his hand as well. Mbuzo, please, you're welcome to unmute yourself and ask your question.
No, I'm sorry, it was a mistake. Sorry, I have no question. Thank you.
Okay. Then I think Muhammad,
Yes.
to ask a question, please. You're most welcome.
Thank you for the lecture. I just wondering, how would you differentiate the usual biophysics and chemical physics that is happening with the quantum biology? And regarding your comment on the the consciousness, what kind of physical or quantum effects that is relevant to that consciousness?
Yeah, thanks. Yeah, as I mentioned, I wasn't going to get into the debate about whether it's just another form of biophysics. To be honest, I think it is just another field of biophysics, but it's one that deals specifically with these interesting quantum effects in biology. So things like superposition that you're not going to find in normal biophysics. But I don't think it's necessarily a completely separate field of research. It falls within biophysics. Consciousness, as far as consciousness goes, I actually will not, I don't have any firm ideas about whether quantum physics contributes to consciousness, but I think one level down is potentially the interesting level at the moment is, do nerves use quantum effects? So if nerves are going to be implicated in consciousness, then does neural function use quantum effects? And as I illustrated at the beginning, nerves are fairly simple when you look at them and compare them to bacteria. And I think, yes, nerves definitely might use quantum effects. So then you might be able to extrapolate that towards consciousness. But I think this gap between the neural correlate of consciousness and what consciousness is a wider question than just quantum biology. It's a question that we haven't pinned down properly across in neuroscience, I think.
Okay, thank you.
Thank you, Betony, for that. And Betony, there is a question in the chat from a biologist perspective. What books, lectures, material should we use to prepare for the next lectures?
There's actually, I can direct you to, we have a useful resources page on the GAF, do science communications for the GAF Foundation, and there's a useful resources page on the GAF Foundation website that has a lot of useful resources. And then Francesco and I wrote a paper on the brain and called Quantum Effects in the Brain, which will take you through the quantum neurobiology that I'm going to look at next week. And then we also wrote a review in the condensed, the Encyclopedia of Condensed Matter that looks at exactly what I'm going to be looking at next week. So the quantum effect and then the biological context.
And we can
thank you. That was my question. Thank you so much for the answer and for the lecture, of course. Just a quick note from a biologist's perspective, of course, those quantum effects and the physics is quite a difficult concept to grasp, especially in such a short time. Therefore, I wanted to ask how to prepare to better understand the whole concept. And also, I would be very thankful if during the future lectures, you could speak about how are those quantum effects studied in the cells, especially because,
Okay.
well, if we have a wet lab, then maybe I can do an ELISA or some cytometry or other kinds of things that I can see to check whether, for example, there is some particle in the or some substance.
That's a great point, actually, because I think what is missing a lot in the conversation between the physics and the biology is that like it's very hard to do experiments in biological systems, especially because you then kill the biological system, or it's just a cell, or it's just even something smaller than a cell. So how can you say it's actually functioning as it would in the real world? So yeah, that's talking about experimental techniques. I think that's a very useful thing to do. Thanks. I'll keep that in mind.
Thank you.
Excellent. And we will definitely post on the NITheCS website, or maybe in the resources of Zoom for next time, the links to the various books, websites, and
yeah, I can put together a resource list as well.
And so on and so forth. There is a question in the chat if we can create a group chat for this. Technically speaking, I don't know at the moment how to do it, but we will explore whether we can use Zoom for an offline chat related to the mini school. To be honest, I wouldn't know at the moment how to do it technically, but we will try. It's a very It's
very
a very,
welcome to.
it's a very interesting suggestion. Yeah. It's just that technically,
And you're very welcome to.
Yeah.
Oh, you're very welcome to email me too if you want to.
Yeah. Maybe Discord or Slack, or maybe Discord is a good idea. Maybe we will set up something. We'll make a plan. But thank you very much.
And I know, I know Omid also wrote a very nice Medium article about.
Yes, he posted the link in the chat, so people should be able to. But we can add that to our collection of resources.
I also want to suggest, if you want to have a closer look at the physics, the summer school was on open quantum systems, which should all be online soon. With
that's
lectures.
also online. No, excellent.
It'll give you a better idea of the tools you need to.
Perfect. Okay, no, excellent. Sorry, I see there are still 2 questions in the Q&A, Betony.
Sorry.
What are the applications of quantum biology in the realm of quantum computing?
Interesting question. I think I'm not sure about other. It was my main topic of interest relating quantum biology and quantum computing is the similarity between, and you'll hear more about this in upcoming lectures. Is there's a model of quantum cognition that uses phosphorus spin in calcium phosphate molecules in nerves, and the argument is that these phosphorus spins are, and you will have seen that phosphorus kept coming up in biological molecules, and phosphorus nuclear spin acts as a good qubit. And what's really interesting to me is that there's a model of quantum computing called the Kane quantum computer that uses phosphorus spin as a way of encoding information in a spin zero silicon substrate. So there are possible ways that quantum biology and these interesting ways in which nature might have enhanced quantum effects with these. I think proteins are really clever materials. In fact, I think quantum biology is basically the study of quantum effects in incredibly clever materials, or incredibly interesting materials. And understanding that, I think, will help with all sorts of things, including quantum computing.
Excellent. Thank you very much. Betony, there's one last question. While wet and messy large systems, the coherence dampens quantum effects. Are there examples of systems which can be usefully partially characterized with such whole system analysis? I'm not sure that I understand 100%. For example, emerging quantum ecology derives some apparently useful entropic and energy models.
I'm not sure I understand the question either, but if you're talking about whether quantum systems can harness the disorder in the environment, then yes, I do think, like utilizing specific protein vibrations. If you want, you can email me as well if you want clarification on.
Yeah, maybe that's better, because I'm also not 100% sure what the question is about. Maybe one last question by Latevi Lawson. Latevi, please, you're welcome to unmute and ask your question, and then we will round up. Latevi, you're welcome to unmute and ask your question. Okay, maybe it was also a falsely raised.
Hello, can you hear me?
Yes, please.
Okay. Yeah, my first question is already asked about the correlation between DNA coding and the quantum information. I think that you already talked about it. My second question is the aging, which is a biological subject, and the decoherence. What is the correlation between them?
The what? What was
the aging?
Aging.
Yeah, and
like
decoherence.
Getting older, like getting older.
Yeah, sure.
I'm not sure if there is a. I'm not sure how decoherence. I mean, the timescales are so different between aging and decoherence, but I do know that aging is a subject of interest in quantum biology because of the effect of reactive oxygen species. So reactive oxygen aging has inflammation has been implicated in aging, and as you'll see, I'll talk about reactive oxygen species more. And there's some thought that quantum principles are operating in the balance of reactive oxygen species. But I'm not sure exactly how deep, unless, I mean, if your protein is aging, you're going to get, aging proteins definitely get stiffer, so their vibrational modes will change. So they might then be slightly out of tune. So that could be a way in which they're, so they're not going to then facilitate electron transfer in the same way if their vibrational modes have changed. Because I know aging is related to stiffening of proteins. So I'm not sure if that answered your question, but.
Yeah, thank you. Thank you.
Okay, then Betony, then since it's just gone 3, Betony, thank you so much for really a brilliant start to the mini school. I enjoyed it really very, very much. So thank you very much from all of us. We will add the links to various things on the website and keep you all all informed. And we are all looking forward to see you again next Wednesday, same time, same Zoom link. Yeah, excellent.
Thanks. Have a good week, everyone.
Again, thank you very much again. Thank you. Thank you very
Bye.
much. Bye. Bye-bye.